Okay, this one will be on proteins. A lot of information to sort of unpack in this section, so I'm going to start with the most basic part of this, and I'm actually going to start with probably the 4 levels of protein structure, because that kind of sets the stage for everything else. So when we're talking about proteins, we're talking about a polymer, and we call that polymer a polypeptide. So poly meaning many, peptide meaning each individual unit. So if I were to break this apart, this would be each individual unit. So there's 5 parts to this polypeptide. Each part of a polypeptide is called an amino acid. So amino acids have a generic shape to them. They have a central carbon first. They have an amine group, an H2 group. They have a carboxylic acid, so COOH. They have a hydrogen, and so those parts do not change. The only part that changes here is up in yellow, and it's called the R group. And the R group determines its characteristics of that specific amino acid. Some of them can be hydrophobic, which means they do not like water, hydrophilic, which means they like water. They can be charged, so it could be positively charged or negatively charged, and those can be attracted to each other, or they might have sulfur. Those are kind of the 5 main ones: hydrophobic, hydrophilic, positive, negative, sulfur. And so, depending on what that characteristic is, for each of these individual amino acids, because they all have their own R group, R, 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 R. The R groups are what change. And so this is what we call the primary structure. It is the amino acid sequence. So in what order are the amino acids? And you'll never have to memorize any specific amino acid sequence or anything, but knowing that that's what defines the primary structure. Secondary structures start to form. We'll see proteins that look sort of like this, or they might look something like this. Other proteins might just be like a garbled mess. So this, and this are all secondary structures. Sorry about that word. That's secondary. Secondary structures, where this is called a beta sheet. Sometimes it's called pleated. Sometimes you might see that word. Here, this is called an alpha helix. So it looks kind of like what you would expect to see with DNA. And then this one is just sort of random coils that interact. So alpha helices and beta sheets are characteristic of the secondary structures for proteins. Then we get into sort of the tertiary. So in between, so tertiary structures would be sort of like how this alpha helice and this alpha helice and then this beta sheet, how all of these, how all interact with one another. And so this gives it its shape. So this is in tertiary structures. So usually we're going to spend most of our time here actually is what is the tertiary structure, because this shape determines its function, right? Remember the lock and key that we've been talking about. Occasionally, proteins will have more than one. I should also note probably in here that this is a polypeptide, right? So there might be an amino acid here, 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 here. These are all amino acid chains. So this one might have, 50 amino acids in it, let's say. What can happen then is you might have one chain that looks like the one I've drawn up here above, and then you might have a second protein that interacts with it as well. And we got some random coils down here. And then we might even say that there's a third one. Say that there's a third one with an alpha helice, a pleated sheet here, and then another alpha helice here. So we have, actually in this case, there are 3 polypeptides, and they interact to form what's called a quaternary structure. This quaternary structure doesn't always exist with proteins. Sometimes they are just limited to a tertiary structure. So if this protein is by itself, up here in the top, that does not have a quaternary structure, it only has a tertiary structure. In each amino acid, go back to this slide here. I'm going to use this one as an example. This protein only works at usually generally specific temperatures and pHs. Those really determine where the protein can function well. So like something that's in our stomach, for example, is going to have a really low pH and a fairly high temperature. And whereas the opposite would be true if this was a protein that was working in our intestines, then the pH would be higher and the temperature would still be higher. If we were to find a specific protein in a bacterial cell growing in some Arctic ice, here's a good example actually. There's proteins that allow plants to be cold weather plants. So their pH range might be sort of more neutral, not high or low, but then their temperature is low. And so it really depends on where do we find the protein. The pH and temperature is going to determine its shape, because what ends up happening is if this protein does what we call denatures, what ends up happening is instead of being in this specific shape that we see up here, it might start to form more of a line structure. So you can kind of see it's lost its shape. And when shape loss happens, then the protein becomes useless. A good example of this, and you see this all the time, is when you cook eggs. So when you cook eggs on the stovetop, that denatures what's called albumin, and we'll look at this in the lab. Denatures albumin and it turns white and hard. And this one is actually irreversible. Sometimes we can reverse this, but it kind of depends on the nature of the protein and how badly we denature it. So I'm just going to draw them as like blocks here. Is a peptide bond. So peptide bonds attach amino acid 1 to amino acid number 2, amino acid number 3, amino acid number 4. So these are all peptide bonds that connect those two to each other. And again, we put that together and we call it a polypeptide. So if I were to last little bit here, I'm just going to go through a little bit of the protein functions. So with these, I'm not going to expect you to know a ton of them. The one that I'm going to really kind of harp on is enzymes, because we're going to spend some time with enzymes. So enzymes are specific. They have that lock and key idea where an enzyme has a specific shape, and this is a protein that has a specific shape, and things fit into the enzyme. And then what ends up happening is sometimes enzymes either Generally, they build things or break down things. And we have something like 20,000 of these, probably more in reality. So enzymes do a lot in our body. They build or break down. We'll focus on the ones that probably break down more in class, but also they can build too. I'm going to give you a handout in class for some of the other ones, but basically it all comes down to shape. So function equals shape. So antibodies on white blood cells, for example, they're going to pick out specific bacteria. So if you have a white blood cell here and it's got a specific shape on it, this is going to target specific bacteria or other pathogens. So that could be funguses. It could be like another person's red blood cells. So like you guys know that you can't cross all different types of red blood cells. So and there's a bunch of them. But basically the thing is to keep in mind is that function equals shape and shape equals function. So if something loses its shape, then its function also gets lost as well, or it changes what its functionality is. I think we'll stop there, and that'll be a good intro to proteins.